this video, we are going to talk about the potential intermediate hosts of SARS-CoV-2 and the transmission of this virus. We will then talk about the SARS-CoV-2 mechanisms and how it replicates within the host. To start with, bats are considered to be the natural reservoirs of the COVID-19 virus. This means that the natural source of this virus is thought to have come from bats. However, bats do not transmit the virus directly to humans, meaning that you cannot get COVID-19 from a bat. Instead, bats transmit the virus to different intermediate hosts, which will then eventually transmit it to humans. An intermediate host is an organism that hosts the virus for a short amount of time, usually in its immature form. It will then pass the virus to a definitive host, such as humans, where it will reproduce. One potential intermediate host could be the pangolin, as the pangolin strain of the virus is 99% similar to the human strain. Therefore, because of the genetic similarity, pangolins could be a possible intermediate host for this virus, facilitating the virus transfer from bats to humans. Some other intermediate hosts that could be possible are minks, snakes, or turtles. But these are not known for sure. Ultimately, the intermediate hosts for the SARS-CoV-2 virus are not definitively known, and more research needs to be done. However, there are some strong candidates that could be intermediate hosts for this virus. Next, we are going to talk about the transmission of the virus and how it spreads. Transmission of this virus can be through respiratory droplets or fomites. Respiratory droplets are transmitted by coughing or sneezing, and then those droplets come into contact with a mucous membrane on a susceptible person, which will then infect them through the respiratory tract. Fomites are objects that the virus can stay on, which will then wait for a human to contact the fomite, therefore transmitting the virus. An example of a fomite is a cup, where if an infected person drinks from the cup, then the infection will be on the cup, and then, if a susceptible human drinks from that same cup, they will be exposed to the infection. Some research has also shown that the virus can be transmitted by aerosols. Aerosols are like respiratory droplets in that they come from a person's respiratory tract. However, they are much smaller than those droplets and therefore can travel a lot farther than the respiratory droplets. These are the main forms of SARS-CoV-2 transmission. However, transmission in children can also be through the fecal-oral route, meaning that they can get infected by eating things that have the virus on them, instead of getting infected through the respiratory tract. Furthermore, it has been found that the SARS-CoV-2 virus can replicate in the intestines of a human, supporting the idea that there can be infection through the intestinal route. Also, while some studies have said that pregnant women who are infected with SARS-CoV-2 cannot pass it on to their baby, there has been a few cases of it happening, and there can be perinatal transmission from a pregnant woman to their baby. However, this is quite rare and needs to be investigated more to be certain. The SARS-CoV-2 virus can not only spread when someone is showing symptoms, but it is possible that it can also spread during the incubation period, which is when someone is infected but they are not showing symptoms. Finally, there has also been human-to-animal transmission, as well as animal-to-animal transmission, including transmission from a human to a dog, tiger, and cat, as well as cat-to-cat and ferret-to-ferret transmission. This has been a review on the intermediate hosts and transmission of the COVID-19 virus. Now we will talk about the replication and molecular mechanisms of SARS-CoV-2.
So how does COVID-19 infect your cells? COVID uses the S1 subunit of the S protein to bind to the ACE2 receptors. And then uses the S2 subunit of the S protein to enable entry into the cell. When it enters, it kind of looks like a bubble within a bubble because it has two lipid bilayers surrounding its genome. After proceeding to pass the first barrier to its entry into the cell, the outer bilayer around it merges with the lysosome, putting the inner bilayer inside the lysosome. You can think of a lysosome like a mini stomach that digests all the food for your cells. At this point, your RNA genome of COVID is still surrounded by a lipid bilayer, which is now inside the lysosome. From here, the inner membrane, or the current lipid bilayer around COVID, now fuses to the wall of the lysosome, like it did in the beginning, except this time, because it is starting from inside, your RNA genome is now expelled into your cell rather than the digestive enzymes in your lysosome. Once the genome of COVID is loose inside your cell, it enlists the aid of an RNA-dependent polymerase to make a template for more of its genome to be replicated in your cell. So it takes the positive sense strand and it makes the equivalent of a negative sense strand, which is then used as a template to make a bunch more positive sense RNA strands. From here, it also recruits ribosomes to translate itself, being the positive sense strand, into multiple copies of proteins, which it then uses to build itself. It then goes through the rough endoplasmic reticulum, or rough ER, and Golgi body to form a new virus that buds out from the cell. The NSPs that were discussed in the first video of this series aid in this process throughout, and are mostly seen in aiding after the genome is inside the cell with replication and in guiding the genome of COVID through the rough ER and out of the cell. Once COVID leaves the cell, it progresses on to infect new cells and start the entire process again. It does this through a cytokinetic storm. This is usually detected through a high NLR, or neutrophil to lymphocyte ratio. This means that it has the capacity to infect more cells, since there are more white blood cells for it to infect, and just more cells in general for it to infect. This increase in cytokines, which are just small proteins that recruit white blood cells to areas, results in basically a bunch of white blood cells being recruited to these areas where your cytokines are being released, which is oftentimes in COVID initially your lungs, which can lead to ARDS, or acute respiratory distress syndrome.